Hello everybody, this is Mr. ForceJ. It's kind of odd talking to you from a computer, but I think this is the way things are going to be done for the foreseeable future. And none of us have any, secret information or anything. We are just like you guys, we're getting it day by day. But we're definitely going to be out for a few weeks, so we need to keep caught up here. The government has said that diploma exams are going to go ahead, but again, that could change. We don't know. Every day we're getting new information. So hopefully everybody's not too panicked. This is kind of the way that I'm going to deliver information to you. We're going to pick up right from where we left off in our genetics. So as far as assessments go, as far as tests and quizzes, I don't know what's going to happen. Again, we're kind of flying by the seat of our pants over here as well. So I don't know if I'll have assignments on the Google Classroom that you'll turn in. We'll have to see. So again, we're kind of all up in the air, but what I need to do is just get you information. Okay, so for the foreseeable future, I will be posting daily lessons. I will go through what I would go through just like if we were sitting in class. Okay, so the last time we were together, I had asked you guys to do some dihybrid questions from your notes. So what I would like to do right now is go through a couple of them that tended to give my classes in the past a bit of a hard time. So this would be on page 8 of your note package. If you haven't done these questions yet, pause this recording right now, go take a look at those questions. Remember, you need to FOIL your gametes in order to get them. You use the appropriately sized Punnett squares, and you should be able to do those, okay? Now, if you have done them, okay, so hopefully you've gone back and done them. If you haven't, if you have done them, continue on. We're going to go through a couple of them here that tend to give people a bit of an issue. Okay, so this one is I believe, number 6 in your note package. Okay, and this one says about 70% of Canadians get a bitter taste from the drug PTC, while other 30% do not. The ability to taste this drug is dominant, while the inability to taste is recessive. Okay, so we want to organize ourselves, and we want to put that stuff down so we know what's going on here. So we're going to say big T is the ability to taste. Okay, you'll have to bear with me, guys. I'm doing this with a mouse at the best of times. My writing isn't that great. Okay, so big T is taste, little t is non-tasting. I know it's painful, bear with me. And then tongue rolling is dominant, so the ability to roll the tongue is dominant. Okay. And then little r is non. Okay, so once we've organized ourselves, we then kind of go and then we kind of have to see what's going on in the question. Okay, so it says here that we have a woman, okay, a tongue rolling woman who cannot taste. So that's one parent. Okay, and then the other parent we have is a man who can taste but cannot roll his tongue. Okay, so those are our two parents. Like the steps that I pointed out to you guys, we need to be able to organize ourselves. Okay, so a tongue rolling woman. Okay, so she can roll her tongue, which means she's definitely going to have a big R here. Okay, we don't know what the other R is, right? Because that's dominant. She could be big R, she could be big R, little r. Okay, we don't know at this point. And she cannot taste. Okay, so she's a non-taster. So that would be little t, little t. Okay. Now, she had a father. Okay, so this woman has a father who could not roll his tongue. Now, that's important information. Okay, because non-rolling is recessive. So if her father was little r, okay, that means she had to inherit a little r from her dad. So we can fill in that blank. Okay, now the man she marries, okay, she marries a man who can taste. So he's a taster, but cannot roll his tongue. So we know for sure that he cannot roll his tongue. That's recessive. So the only way to be recessive with Mendelian genetics is to have the two recessive alleles. Okay, but he can taste. Okay, so he's a taster, which means it's a big T, little t. Oh, actually, we don't know. Okay, he can taste. So sorry, let me just erase that. Okay, we don't know at this point. So can we find out though? Well, if we look in the question, we can see that his mother was unable to taste. Okay, so that means that his mother had to be little t, because that's the recessive form. So his mother would have given him one of his alleles, right? That's Mendel's first law. You have to get one allele from one parent, one allele from the other parent. Okay, so now we know what the genotypes of the parents are. Our next step is we need to FOIL out the gametes. Okay, so big R, little t, big R, little t, little r, little t, little r, little t. Okay, so this woman can only make two types of gametes, a big R and a little t, and a little r and a little t. Okay, now the husband, now again, if you wanted to do a 4x4 Punnett square, that's fine, right? But you guys know my motto, work smart, not hard. So you would just be doubling your results, right? But that's fine if you want to do it. If you want to FOIL out all four here, you'd have two repeating gametes. I just got rid of the repeats, okay? But if you wanted to do it by 4, that's fine. Now, the man she marries, little r big T, little r little t, little r big T, little r little t. Okay, so he can make two types of gametes, little r big T, and he makes a little r little t. Okay, so again, like the mom, like the woman, the man can also make four type. If we FOILed it out, we would get four gametes here, but only two different types of gametes. Okay, so I'm basically cutting my work in half by getting rid of those gametes that repeat. All right, so now we put them back together again in the Punnett square. Yeah, I know this is probably, it's as painful doing this as it is watching it. I wish I had a pen. I'm just doing this with my mouse. So it's going to take me a bit to get used to this. Hopefully, in later presentations, it'll go a little smoother. Okay. Little r, little t, little t. Okay, so the question says, use a Punnett square to illustrate your answer. Provide the genotype and phenotype ratios of the offspring. Okay, so genotype ratios, we have roll and taste. Okay, so one roll and taste. We have a one roll and non-taste. We have a one non-roll and taste. And we have a one non-roll and non-taste. So both the genotype and phenotype ratios of this cross would be a 1 to 1 to 1 to 1. Now, if you did it the long way, you would have got a 4 to 4 to 4 to 4 ratio, but you would have had to reduce it to a 1 to 1 to 1 to 1. Okay, so this one is number 7 in your notes, and this one is quite a long question. It requires a lot of work, so we'll go through it, and I'll see if I can kind of get you the idea of what they were trying to ask in this question. So a botanist is trying to create a pea plant that is homozygous for green pod color and round shape. Both of these genes are dominant over yellow and wrinkled. Okay, so again, more Mendelian stuff here. So we have to organize ourselves. Always, guys, always organize ourselves. Okay, so we want green. Big G is going to be green. Okay, big G green. Little g is yellow. Okay, and we have big R is round. And little r is going to be wrinkled. Okay, so again, whatever letters you use is fine. If you used, big C for green color and little c for yellow color, or big S for seed shape or whatever, right? I mean, these kind of make more sense, but the letters don't matter. Okay, so little r for wrinkled. Okay. The botanist has collected a group of pea plants that produce round peas and green pods. Okay, he crosses these plants with a group of plants that have wrinkled peas and yellow pods. So right away, well, there you go. Some announcements interrupting class. Now you guys all feel like you're actually at school. Okay, so what we want to do is we want to follow our steps, and we want to put down the genotypes of the parents. Okay, so the botanist has collected a group of pea plants that produce round peas and green pods. Now that's the dominant phenotype. So we don't know what the genotype is, right? So that's an unknown. We know that they have a big R. We don't know if they have a little r or not. And we know that they're green, so they're going to have a big G, but we don't know what the other letter is. Okay? Now, he crosses these plants with a group of plants that have wrinkled peas and yellow pods. Now, some of you, well, maybe, I don't know, maybe your brains are cramped a little bit right now because you haven't thought genetics in a while, but we're crossing this unknown with a known genotype. So we know for sure that the wrinkled yellow pea is going to be little r, and for sure it's going to be little g. Okay? So does that ring a bell for anybody? Do you know what this is called? Anyone? Right? When we have an unknown and a known, this is called a test. Hopefully you guys are figuring it out quicker than I can write it. Test cross. Okay, so we have our test cross. All right, so show all the possible combinations that may result from this cross and provide the phenotype ratios for the offspring. All right, so this is where it gets really long, okay? Because basically what they want you to do is come up with every combination of round and green. So we could have big R, we could have big G, okay? We could have big R, little r, big G. We could have big R, big G, little g. Okay, or we could have big R, big G, little g. So all of those plants are round and green. Okay, that's all possibilities for round and green. So what they wanted us to do, and we would have to actually do 4 individual Punnett squares crossing with this unknown and figure out what the phenotype ratios are. Okay, so if you want to do that, great, go ahead. If you need to practice, fantastic. So basically you take big R, big G times little r, little g. What would all the offspring be? Well, they'd all be heterozygous for both traits, right? Then you would take this one and times it by this one, and then you would take this one, times it by this one, times it by this one. Okay? Now, again, we can do that, or we can just kind of take a look and see if we can figure out some patterns. Okay? Now, the botanist determines that the phenotype ratio of the offspring is a 1-to-1 ratio. We have 1 round in green to 1 round in yellow. Okay, so that tells me something. It tells me something right away that one of my offspring is yellow. Okay, now why? What does that matter? Well, what that tells me is in order to get yellow, I have to have 2 little g's, right? Because that's recessive. So I'm going to get 1 little g from this parent right here. Okay, but where am I going to get the other little g? So one of my offspring is yellow. Okay, so one of my offspring is going to have to be little g. Okay, it's one of the offspring. The other offspring doesn't tell us anything, but this offspring tells us that it's got to have two little g's. So where is it going to get its little g's from? It gets one from this parent, but that means it has to get one from this parent. Okay, so we know right away that we can cross this one off. That's not one of the parents, and we can cross this one off. That is not one of the parents. Okay, now the fact that all the offspring, okay, have round seeds. Okay, right? One of them is round green. Okay, what was the genotype? Oh, sorry. One of the offspring is round and green. The other one is round and yellow. So we have no wrinkled seeds there. So that rules out a parent with a little r, right? Because if you have a parent with a little r and you have another parent with a little r, some of them are going to be wrinkled. But the fact that none of the offspring are wrinkled tells us that our parent is going to have two big r's. Because if our parent has two big r's, then there's no way to ever get offspring that are wrinkled. Okay, so again, all of our offspring are going to be round, but they're going to be carriers for the wrinkled allele. Okay, yeah, and then some of them are going to be big R, little r, and some of them are going to be big G, little g. Okay, so if you did a Punnett square between these two parents, what you would find is that you would get a 50-50 outcome. 50% would be round and yellow, and 50% of them would be round and green if we had this parental type. Okay, the other way to do it is to actually do every single one of these parents, all four of these unknown parents, cross them with our known, look at the Punnett squares, see what you come up with, and then you would figure out which one gives us our 50-50 cross. Okay, the other way is just to kind of logically look and say, what does my one parent need that my other parent doesn't? Okay, all right. The next part of genetics involves some traits that Mendel didn't see with his pea plants. So remember, he used pea plants because they were relatively easy to grow. He could self-pollinate them to make sure that he had, his pure breeding garden of green pods and his, pure garden of pure breeding purple flowers and all that kind of stuff. So he stuck with pea plants. Okay, but there's lots of different organisms in this world other than pea plants. And it turns out that some of them don't follow Mendel's laws. So here I have an example of a type, okay, called a snapdragon. And in snapdragons, when we take a pure breeding red flower and we cross it with a pure breeding white flower, okay, we don't end up with red. Let's say that, red is dominant, right? If this was Mendelian genetics, let's say red is big R, right? And white is little r. What we would expect is 100% of our F1 generation to be red. But in this case, when we cross pure breeding red and pure breeding white, we get something completely different from either of the parents, and we get pink flowers. Okay, so it's not red. You can see in the picture it's not red, it's actually pink. So the heterozygous condition here is not red like we would expect with a pea plant, but with snapdragons, it's actually something different than either of the parents. Okay, and this is what's known as incomplete dominance. So red is not totally dominant over white. It doesn't completely mask white. Okay, what it does is it kind of blends with red, and white kind of blends with red. The two alleles talk to each other on the two chromosomes, and we get something kind of in between. Okay, we get a pink. Now again, if we did the Punnett square for this, all right, so this is our red snapdragon. Okay, this is our white snapdragon. And what we end up with is we get 100% of my F1 generation. Okay, 100% of the F1 is not red like we would see in Mendelian genetics, but it is pink. Okay, so once we have that third phenotype, so white is one phenotype, red is one phenotype, pink is a third phenotype, that tells us this is non-Mendelian genetics. Okay, it does not follow Mendel's first rule where he says we get 2 alleles, 1 from each parent. Okay, that's great, that's fine, but he said that we have dominant and recessive. And in this case, we do not have dominant and recessive. We have incomplete dominance. Okay, now what would happen if I took 2 of these pink flowers? Okay, I take this pink flower and I take this pink flower and I cross them. Okay, what would happen in my F2 generation? All right, so the allele R still codes for red, the little r still codes for white, but in incomplete dominance, heterozygotes are phenotypically distinct from the 2 homozygous varieties. So homozygous red, homozygous white, heterozygous is pink. That's phenotypically distinct. Okay, so now here I have 2 pink flowers. Okay, those are my 2 F1 flowers. And I cross them together. What ratio do I get? If you'll recall with Mendel's flowers, if you took a purple and a white and crossed them together, you got 100% purple. Then if you took 2 of those purples and crossed them together, you got a 3-to-1 phenotypic ratio, purple to white. But what do we get here now? This guy is red. So this is F2. These are my F2 plants now, right? Because I crossed 2 F1 plants. It's an F2. Okay, that's red, pink, pink, and white. So I don't have a 3-to-1 ratio in my F2 generation like I did with Mendelian genetics. What I end up having is a 1-to-2-to-1 ratio. Now people always ask, okay, which one do I put first? Why is this not a 2-to-1-to-1 ratio? Or why isn't it a 1-to-1-to-2 ratio? Well, we always start with the most dominant, okay, with the most dominant alleles. So, okay, this guy has 2 dominant alleles, so that's our first ratio, part of the ratio. This one has 1 dominant allele, so it goes next, okay, and this one has no dominant alleles, so it goes last, all right? So this is a phenotypic and genotypic ratio, 1-to-2-to-1, okay? Red to pink to white. All right. Now, we do have this in humans as well in a few different places. This is an odd disease. It's called hypercholesteremia. And for you guys that don't remember, in Bio 20, there's 2 types of cholesterol, right? There's HDL cholesterol and there's LDL cholesterol. LDL is quote unquote the bad cholesterol. I mean, it's used in our body, but if we have too much of it, it can plug our arteries and it can kill us. Now, LDL cholesterol, we typically get rid of in our livers. Okay, so some people can deal with LDL cholesterol very easily, some people can't, and it's mainly genetic. Okay, to be honest with you, they used to think it was a lot to do with diet and that stuff. I mean, eating less cheeseburgers can definitely help your body have less LDL cholesterol, but it's mostly genetic. Okay, so there's a gene that deals with the breakdown of cholesterol, and if you are actually homozygous recessive for this gene, so if you're little h, you're normal, okay? Which means that your body can deal with LDL cholesterol and there's no increased risk of cardiovascular disease, okay? So if you eat healthy and you exercise and you keep a good weight, you don't have to worry about clots in your arteries as much, okay? Now, why am I talking about this disease? Well, because what we find here is that if you have a big H, little h, okay, this is different than this. So heterozygous is different than homozygous dominant. And in Mendelian genetics, these two would phenotypically be the same, okay? Meaning that there would be no difference between big H, little h, and big H, big H. But here we have a difference. So if you're heterozygous for this disease, then LDL levels are abnormally high, okay? They can be treated with drugs called statins, okay, which can reduce your LDL levels. But if left untreated, people can have heart attacks and strokes by their mid-40s, which is early, okay? So if you're heterozygous, you have increased risk. If you're homozygous recessive, you're normal, no increased risk. And then we have a third phenotype, okay, big H, big H. If you are big H, this is real bad news. And you can get heart attacks and strokes and stuff by your early teens. And typically, if left untreated, death can occur in the early 30s. So here we're dealing with 3 different genotypes. But we also have 3 different phenotypes. We have totally normal, we have kind of increased risk, and then we have majorly increased risk. So because we have 3 phenotypes, this is non-Mendelian, okay? Now, sickle cell anemia is another one. Sickle cell anemia is a disease that some humans get. And again, it's caused by a faulty gene. So in our red blood cells, we have a protein called hemoglobin, and hemoglobin carries oxygen, okay? So if we have the normal genes to make normal hemoglobin, then our red blood cells are nice and round, what we call biconcave. They look like a disc, and they flow through our body very easily, and they carry lots and lots of oxygen, okay? Now, if you are born with the recessive, homozygous recessive condition, then your blood cells look like a crescent moon, okay? Now, a sickle was an old type of instrument that they used to cut down crops and stuff, and it had this curved blade to it, okay? And that's kind of what these cells look like. Now, there's a third phenotype, okay? So again, homozygous dominant, totally normal. Homozygous recessive, totally abnormal, crescent shaped. But if you're heterozygous, it's a little bit different from either, okay? So if you are heterozygous, so big S, little s, you don't get sickle cell, but you have a mutation in your hemoglobin that does not allow malaria parasites to get into your bloodstream. Okay, so malaria is carried by mosquitoes. There are high rates of malaria in warm countries like Africa and South America. Okay, there are some African countries where up to 40% of the population has sickle cell, because the reason for this is because the heterozygote condition is actually an advantage. So if you're big S, okay, you don't get sickle cell. You can carry, your blood cells are normal, but they have a high incidence of malaria, okay? If you're homozygous recessive, little s, then you can die of the anemia that's caused by the sickle cell disease. It's very painful from what I understand. You get blood clots and you get anemia, so you would die. So again, you die from malaria if you're big S. You die from sickle cell if you're little s. But if you're big S, little s, then you don't die of either. And this is called the heterozygote advantage, okay? And because most of the population in some of these African countries is big S, little s, okay, that means that, the chances of meeting someone else that are big S, little s are much higher because of evolutionary pressures, right? Most people in the community with high malaria are going to be heterozygous. That means that almost every couple is going to have a 25% chance of having a kid with sickle cell disease, full-blown sickle cell disease, okay? The other kids that don't have sickle cell disease, they would die of malaria. So you have a much higher incidence in these countries of people that are both carriers of the sickle cell trait and who have actually sickle cell disease, okay? So some populations have 10 to 40% of their whole population has sickle cell disease, because again, this the heterozygote genotype gave their parents an advantage over malaria, so they would continue to have kids at a higher rate with sickle cell. Now, another non-Mendelian trait is known as codominance. And in codominance, both alleles are fully expressed. Okay, so this is different than incomplete dominance. Remember, in incomplete dominance, a red plus a white equaled a pink. Here, with codominance, what we have is we have both alleles being expressed at the same time. Okay, so as in this picture here, you can imagine that this flower might have had parents that were white and red. Okay, and both white and red are being expressed at the same time rather than a blending of the two. Okay, so a special type of codominance is known as roan. Please make a note of this word. Okay, roan means that two different alleles are being expressed at the same time. Now, if you were to look at this horse and tell me it was gray, okay, I would actually tell you you're wrong. It's not gray. Okay, take note of the head here. Take note of the ears and the mane. That's white and black. So this horse actually has two different types of hair, white and black. In the body, both of those colors are expressed at the same time. So this is not gray hair, this is white and black hair. Okay, so that would be codominance, right? If it was white plus black equals gray, that's incomplete dominance. But here we have white plus black equals white and black. Okay, so that is known as codominance. So let's try this one together. Okay, so for shorthorn cattle, the mating of a red cow with a white bull produces a roan calf. So what that's telling me is roan is a special word. It's like test cross. You need to know what it means. Okay, you need to know that it means both traits are being expressed together. Okay, so here I have my white bull. How do I know it's a bull? Well, you guys should probably know that, right? That's the cow over here. Well, that's another biology lesson for another time. But yeah, this is my red cow, okay? And the red cow has, two red alleles. So we can't use big R and little r anymore because there's no dominant or recessive. So what I'm going to do is I'm going to say, okay, you know what? This cow has a color allele. Okay, the cow is red, so the cow is going to have two red alleles. All right, so what I'm doing here, this is the same as saying this cow is big R, big R. Don't get confused because I'm using two letters here. Okay, this is one allele, but I can't say big R anymore because big R is, there's no dominant, they're co-dominant, right? So what I mean by that is that a big R, little r is meaningless here because there's no dominant and recessive. So what I'm doing is I'm just using a different type of notation to show that I have a different type of relationship between the alleles. Okay, so again, you can use whatever notations you want. I find it easier to say, okay, C means color and R means red. Okay, and then the other allele is C for color and red. That's how I get a red cow. Now, how do I get a white bull? Well, I have the color allele, but the bull has white. Okay, and in order to be white, I have, again, that bull has to have both white alleles. Okay, otherwise it wouldn't be white, right? It would be something else. So in this cross, what I'm doing here now is, again, like every other genetics question, I have to make my alleles, right? So this is one parent. And again, I'm not foiling anything here, ladies and gentlemen. Please don't foil because these aren't two different traits. All I'm dealing with is just the color. I know I'm using two letters, but that's because I can't use big letters and little letters anymore because there's no dominant and recessive. So what I'm saying here is, okay, this is my red cow. So one ova can carry red instructions, and actually that's all that this cow can carry is just red instructions. And then my white bull can only carry white instructions. Okay. So when these letters get together, I don't put the two C's together. Again, guys, these are not two separate traits. This is just color. There's no foiling involved here. Okay? So all I'm showing here is that, okay, I'm getting two alleles, one from each parent. Right? Getting one from each parent. And what that's showing is that the calf, okay, every calf actually from this pairing is going to have a C with an R, okay, and a C with a W. So what color are the calves going to be? They're going to be red and white. And what do we say when both colors are expressed? Okay, in animals, we say that this calf is going to be roan. It's both red. This is my C with the R. Oh boy. Okay, and this is the C with the W's. Okay, so both are being expressed here. That's known as roan. Okay, and that's codominance. Yeah, I don't know. I found this online, and because I'm a bio nerd, I kind of found it funny. But we have a dad with stripes that go this way, and we have a mom that stripes that go vertically, and then the kid has both stripes. So what type of genetics is this? I don't know. It's like I said, it's kind of a nerdy thing, but I found it funny. That's codominance, right? Because both are being expressed. So both the horizontal and the vertical lines are being expressed in the child. Okay, so that's codominance, different than incomplete dominance. Now, multiple alleles. Again, this is non-Mendelian genetics because in Mendelian genetics, Mendel said that there were only two forms of the allele. Right? There was dominant and recessive. Now, it's important to understand that you can never have more than two alleles. Okay, because we are diploid. So we have two chromosomes, one from mom and one from dad. So we're always going to get two alleles, all right? But where Mendel made a mistake, and it's not a mistake, it wasn't his fault because in his pea plants the case was there were only two types of alleles for each trait, right? Round or wrinkled, yellow or green, purple flowers or white flowers, or tall or short plants or whatever, right? So he only had two conditions for each trait. But in humans and in other animals and other plants, that's much too simple. So what I want you guys to understand is that we'll only ever get two alleles, but sometimes there's more than two alleles to choose from. Okay, which is the case for many traits out there. Okay, and Mendel didn't take this into account. Classic example is fur color in rabbits. Okay, so we have white rabbits, we have Himalayan rabbits, we have chinchilla rabbits, and we have these dark gray rabbits. Okay, so how the heck do you get four different colors in these rabbits? Well, the answer, and Mendelian genetics could not answer that. So for us, we understand now that there are more than two alleles for some traits. Okay? And in rabbits, that's the case. So for example, if you have just a big C, that's the allele for dark gray. If you have a C with a CH, that's light gray. Okay, if you have a C with an H, that's white with black points. That's known as Himalayan. And if you have a little c all by itself, that's white. So there are four different alleles. Now, if you were to make a rabbit, okay, you're not going to take all four of these alleles. What you're going to do is you are going to choose from two of them, right? Because a rabbit can only have two of these alleles out of this pool because they only have two chromosomes, one for each parent. So how many different ways are there to be dark gray? Well, here's the thing. Not only is there more than two different alleles, but these alleles fall into something known as a dominance hierarchy. So big C is the most dominant. It's dominant over everything else. So how do we get this dark gray rabbit? Well, it's going to have a big C, but it could also have a C with a CH, right? Okay. It could be big C, big C. It could be big C and a C with an H. Okay. And it could also be a big C and a little c. So all four of these genotypes are all dark gray. Okay. And what I want you to understand here is that anytime I wrote out genotypes, I only had 2 alleles, but there are 4 alleles to choose from. Okay, now what's the only way to get this albino rabbit? That's the only way to get this albino rabbit, little c, right? Because if I had anything else, then it would not be white, because that's the least dominant of all the alleles. So this is much different than Mendel's 2 alleles, 1 dominant, 1 recessive. Here we have 4 alleles. Some are dominant, more dominant than others. So this is not Mendelian genetics at all. Okay, this is not simple genetics. So let's try an example here. Okay, so a chinchilla rabbit has a genotype of CCH, okay, CCH, and C with an H. Okay, and it's crossed with a Himalayan rabbit. And the Himalayan rabbit is thought to have the genotype of C with an H, little c. Okay, what is the expected phenotype ratio of the F1 generation? All right, so I have my 2 parents. Now I need to make my Punnett square. Okay, here's my Punnett square. And now this parent, I'm not foiling anything. Remember, these are not 2 traits. It's just that I'm not using big C's and little c's anymore, because that doesn't work, right? Because I have more than 2 alleles and one isn't dominant to the other. You know, it's very messy, so I'm using a different way of doing things. So there's no foiling here, okay? One allele is C with a CH, okay? And the other allele is a C with an H. That's from the 1 parent. That's the chinchilla parent. The Himalayan parent can make a C with an H, and it can make a little c. Okay, what is the expected phenotype ratio? So that's what do they look like? So I have a C with a CH and a C with an H. Okay, that's 1 chinchilla. C with an H, C with an H. That's a Himalayan. I have a C with a CH and a little c. That's still chinchilla, because chinchilla is dominant, right? Then I have a C with an H and a little c, and that would be Himalayan. So my phenotype ratio would be 1:1 chinchilla to Himalayan. It's actually let's not be lazy here. It's a 2:2 ratio, but remember, we always have to simplify, so it would be a 1:1 ratio. Okay. Now, in the above cross, some of the offspring turned out to be pure white. Obviously, the given parental genotypes were wrong. What must be the genotypes of the parents to get albino? So the only way to get albino, because it is, excuse me, it is the most recessive of all the alleles, the only way to get albino is to have 2 little c's, okay? Which means that each parent has to give a little c, okay? So one of the parents was Himalayan, so it has to have the C with the H, but it can also have a little c. Okay, the other parent is chinchilla, so it has to have C with the CH, but it can have 2 alleles here. It can have the C with the H like they thought, but in order to get the albino, it has to have a little c. Is this rabbit still chinchilla? Yes. Yes, it is still chinchilla. Is this rabbit Himalayan? Yes, it's Himalayan. There are multiple ways to get chinchilla and Himalayan, but if we're going to get a rabbit that's albino out of this cross, then both of these rabbits have to carry the recessive allele. Okay, so obviously they were wrong with their genotype when they said it was CCH, C with an H. It has to have a little c. Now, human blood type is another example of non-Mendelian genetics. Here we have a bunch of stuff going on. Mainly, we have multiple alleles. So instead of with human blood typing just having a big A and a little a, because we have 4 different types of blood types, Mendelian genetics does not work here. So we actually have 3 alleles for human blood typing. Okay, we have IA, that gives us A-type blood, IB gives us B-type blood, and little I, with no letter, is no antigen. So again, back to Bio 20, if you'll recall, how do we get the 4 different types of blood types? Well, it's because of these little markers that are on the surface of the blood cell. So if you're A-type blood, that means you have an A marker, okay, and that's coded for in your DNA, right? If you have a B-type blood, that means you only have the B marker, and again, that's coded for in your DNA, right? If you are AB blood, that means you have both markers. You have the A marker and the B marker, and if you're type O blood, that means you have no markers, okay? So there are 4 blood types, and obviously this does not follow Mendelian genetics because we have the expression of 2 of the markers here. We have only 1 here, and none here. So something else is going on there, right? So what's going on is we have multiple alleles. There's 3. Okay, there's the A allele, the B allele, and the O allele. And A and B are co-dominant, meaning they are expressed together. Okay, so in order to have AB blood, that would mean you have to have an A allele and a B allele, and they're expressed together. Okay, in order to have type O blood, that's the phenotype, you have to have both the little i's here with no marker on it. Okay, so either of these alleles have the information to make a protein marker. Now there's 2 different ways to be type A blood. You can get 2 A alleles, or you can get 1 A allele and the other one have no information on it. So A and B are co-dominant, but A is dominant to O, and B is also dominant to O. So we got a lot going on here. Okay, so again, to be B blood, you can either inherit both the same alleles, so that's still known as homozygous, or if you're heterozygous, one of the markers is for B and one of the markers is for nothing. B is dominant to nothing, so you would still get the B expression. Okay, so multiple genotypes, 4 phenotypes, and there's a lot going on here. Okay, so an example. If a man has type AB blood and a woman is heterozygous for A, okay, now they have to tell us that because there's 2 ways of being A. What are the possible blood types of their children? So, okay, our man has type AB blood, so that means he has both the A allele and he has the B allele, okay? And the woman is type A blood, but they told us that she's heterozygous A. Because remember, there's 2 ways to be A. So she definitely has an A allele, okay? She could have another A allele and still be type A blood, but they tell us she's heterozygous. So she has the allele for nothing, okay? So she's still going to express A because it's dominant over this one, but she's heterozygous. So what types of children can they possibly have? Well, we just do a regular old Punnett square like we've been doing the last little while. Okay, so our guy here, he makes 2 types of sperm. He makes an A and a B. And our gal here, she makes 2 possible ova. She makes one with an A and one with nothing. Well, it's still an allele, but it codes for no protein. So we can get type A blood. Okay, so their child could have type A blood. Their child could have type AB blood. Okay, their child could have type A blood, or their child could have type B blood. So I'm being lazy here, guys, just because this mouse sucks to write with, but I should be putting the little eyes there, right? Or those eyes. I am being lazy, I'm sorry. But yeah, this thing sucks to write with. Okay, so again, what possible blood types could their children have? Well, there is a 50% chance that the child will have type A blood. there is a 25% chance that the child could be AB, and there's a 25% chance that the child could be B blood. If the mother of a child is blood type O and the child is blood type A, what possible genotypes could the father be? Okay, so if the mother of a child is type O, so mom over here, she is type O blood, so she's going to have these two little eyes, right? Now the child is A blood, so the child is going to be an A, and the child is going to have a little O. How do I know that? Well, because that's all mom has to give. So what possible genotypes could dad be? Okay, so mom is going to give this allele, okay, right? Because that's the only allele that mom has to give. Now, what about dad? Dad has to give an A, right? So could dad be big A, big A? Yep. Could dad be A with the O allele? Absolutely. Could dad even be AB? And the answer is, yeah, for sure, because dad's going to give one. We don't know which one. Maybe dad gives B next time, right? But this time, dad gave A, and the child has type A. Now, could dad be type B blood? And the answer is no. Okay, dad could not be type B blood. Even if dad was I with a B and a little I like this, okay, what if dad gave this one and mom gave one of hers, then the child would be O, right? What if dad gave this one and mom gave one of these? Well, then the child would be B. There's no possible way that dad could have this blood type. And in the old days, before Maury and his "you're not the father," shows that where he did DNA evidence, this is kind of how they proved paternity, was they would do blood tests. So if mom was type O and the kid was type A and dad was type B blood, well, that's impossible. It's genetically impossible for that man to be the biological father of that child. Okay. Now, also found on blood cells is a different type of a protein called a rhesus factor. Now, the rhesus factor is named after a type of monkey, a rhesus monkey that this was actually found on. So it's different than the ABO blood typing proteins. Okay, it's completely different. It's found on a different chromosome, and it's not taken into account with the ABO blood proteins. Okay, so when we're dealing with this, it's like dealing with two separate traits. Okay, we're dealing with ABO blood typing, that's on one pair of chromosomes, and then the rhesus factor proteins is on another set of chromosomes. So it's no different than saying, remember our examples with the chocolate lab and the PRA, right? The blindness. So we have code color and we have whether you can see or not. So that's two different traits, and that's a dihybrid cross. So just because these are two blood proteins doesn't mean that they're related. They're actually not related at all. Okay, they're two separate traits. So we have to remember that when we're doing crosses. And we actually have to do dihybrid crosses here, and we have to do foiling. Okay, so things get a little bit crazy here. And I have to warn you, that the diploma does like to ask these blood typing questions. Okay, so this is something in your notes you might want to highlight. Again, I don't know if the diploma is a go. It is as I'm sitting at my desk right now. It's 2:00 on Tuesday, the 17th of March. This is diploma's a go. Okay, I'm hoping that changes, but right now it is the way it is. Okay, so what do we do here? Well, we're dealing with two things. We're dealing with rhesus factor and we're dealing with ABO blood typing. Okay, and it tells us that a man is type O and heterozygous positive. So O positive. That's the genotype. Okay? O positive. I'm sorry, that's not the genotype. That's the phenotype. We have to figure out what the genotype is. Okay? So the man definitely has to have two little alleles for blood type. Okay, now it tells us that he is heterozygous for positive. So what that's telling me is that, okay, what we're going to say is that big R is positive and little r is negative. Okay, because this is a Mendelian trait and it's just dominant and recessive. Okay, so they're telling us he's heterozygous for positive, so he would be a big R and a little r. So that is how you would write O positive. Now again, remember these are two separate traits, so I have to FOIL that out later. Okay, now the woman has AB negative. Okay, so the woman's genotype has to be A, B, because those two traits are co-dominant. So to be AB, she's got to have both of them, and she's negative. Now that's the recessive trait, so for rhesus factor, so she has to be like this. Okay, so that would be the genotype for AB negative. Okay, what is the probability that their child has type B negative blood? So we have to do a Punnett square, but in order to do that, because we're dealing with two separate traits, we have to FOIL out our gametes. Okay, so dad can make two different types of gametes. He can make a little I with a positive, and he can make a little I with a negative. Okay, now mom also, we have to FOIL out her gametes. Okay, so she's capable of making two types of gametes. She can make an IA, and she can make an IB, and she can make it with only little r's. Okay, because she's negative and that's all she carries. So again, guys, I'm going to remind you, I am doing the shortcut method here. If you wanted to FOIL out and have 4 gametes across here and 4 down here, you're going to come up with the same ratio as me, just it would be times 4. Okay, so we get the IA, little I, big R, little r. Okay, we get the IA, little I, little r, little r. Sorry, this is an A. Okay, we get the IB, little I, big R, little r. And we get IB, I, little r, little r. Okay, so again, if you did this, you would have just repeated this 4 times. Okay, if you did this the long way, you would have just repeated what I've done here in the 4 quadrants. So they want to know in the question, what is the probability that the child will be B negative? Okay, so if I look here, this one right here is A positive. Okay, this one right here, A negative. This one right here, B positive. And this one right here, B negative. So the probability of having a child with B negative blood would be 25%. Okay, or 1 out of 4 or 0.25 or whatever. Okay, so this is the only outcome that you get. All right. Now, the last non-Mendelian genetic inheritance pattern that I want to go through today is called polygenetic inheritance. And it's a mode of inheritance in which 2 or more genes determine the phenotypic expression of a trait. Okay, so that's a lot of science talk, but basically what they're saying is instead of just one gene for eye color, like a lot of people, think that, oh, you can either get, big B is brown, dominant, little b is blue. Well, how do we get green eyes? How do we get hazel eyes? How do we get gray eyes? How do we get really light blue eyes? How do we get, dark blue eyes? There's more than one gene involved. Skin color, hair color, same thing. So there's for skin pigmentation that we know of now, there's actually 3 genes involved. So there's an A gene, okay, there's a B gene, and there's a C gene, and all 3 of these genes together determine one trait. Okay, A doesn't, talk about hair color, B doesn't talk about eye color, and C doesn't talk about skin color. No, all 3 of these genes all determine skin color. Okay, so in this way, if you're little a, little a, little b, little b, little c, you'd have the very lightest skin. If you were big A, big A, big B, big B, big C, you'd have the very darkest skin. And depending on how many big letters and little letters you have, you fall somewhere in the middle. Okay, so this is known as poly. Poly means many. So many genes are involved in this trait. So here we have an instance where we have 2 different genes dealing with the trait eye color. So it's not as simple as some of the questions that we've been doing in the homework and stuff you've been reading, the real simple big B is equal to brown, little b is equal to blue. We used to think that was the case, but now we know that's definitely not the case. There's more than one gene. There's 2 in this example. I've read that they've now found a third. But basically the way it works is we have the B gene, okay, and we have the G gene. So B and G together determine your eye color. So the more big letters you have, the darker your eye color is. The less big letters you have, the more recessive letters you have, the lighter your eyes are. Okay, so big B, big G. This would be an example of the darkest of eyes. A little b, little g would be the lightest of the eyes. So a big B, little g, big B, little b, big G, little g, that's kind of in the middle. That might be your hazel, right? If you have 2 little b's, okay, and you have a big G, that's how we get green eyes. So here you can see little b, big G, that would be the greenest of the green, right? And then big B, big G, little g, again, here's a little bit more, lighter, right? More toward the blue. And then finally, little b, little g, we get very blue eyes. All right. So is it possible for 2 brown-eyed parents to have a blue-eyed child? And the answer is definitely, right? So if we had a big B, big G, little g, times big B, big G, little g, okay, if this parent gave this B and this parent gave this B, we'd have little b. Okay, if this parent gave this G and this parent gave this G, we'd have little g. So could 2 brown-eyed parents have a very blue-eyed child? And the answer is yes, if they were carriers for both the little b and the little g. Okay. So here we have a special type of polygenetic inheritance called epistasis. And in epistasis, we have 2 genes that dictate one trait. Okay, so for example, the trait I'm talking about is flower color, and this is a type of flower called a sweet pea. Don't get it confused with Mendel's peas, they're completely different. Okay, so in a sweet pea, we have flowers. They can be purple or white. Okay, so purple is the dominant gene, white is the recessive gene. And just like in Mendelian genetics, purple, big P is purple, big P is purple, and little p is white. Okay. Problem is we have another gene that interferes with the color gene, the purple gene, and it's called the control gene or the permission gene. It's like a switch. So in sweet peas, flower color is controlled by 2 genes. In the first gene, purple is dominant over white. But there's also a second gene that allows the expression of the flower color gene. Okay, so it's the switch gene, it's the permission gene. If the plant has a big C, okay, so big C or big C little c, and the purple gene has permission to express itself. If the plant is little c, so homozygous recessive, then the purple gene does not have permission to express itself, and the flower will be white anyway. Okay, so what does that look like as far as genotypes go? Okay, so if I have big P, okay, that's purple. Okay, now the other way to be purple would be to be big P or little p. So either of these genotypes is purple. Okay, now like regular little p in regular Mendelian genetics, when we have just a dominant recessive, this would be white. Okay, but unfortunately it's not that simple. We have a second gene that's involved. Okay, so if the second gene, which we call the control gene or the permission gene, if it's big C, or if it's big C, then this gives purple permission. Okay, permission to express itself. So what I mean by that is if I have big P, that's purple, and I have big C, okay, this gives this permission to express itself, so this would be purple. Okay. Now, what if I had big P, which is purple, right? But I had little c. Okay, now this condition right here does not give permission. So what it would do is it would go back and it would interfere with the purple, and this would now be white. Okay, so little c is like a switch. Little c switches purple off. All right, now let me ask you one more question. What would little p, big C be? Okay, so little p, big C. So the first thing we have to ask ourselves is, does the plant know how to make purple? And the answer is no, the plant doesn't know how to make purple. Okay, the second thing we have to ask ourselves is, if it did know how to make purple, does it have permission? And the answer is yes, it has a big C, so it has permission, but it does not know how to make purple, so this plant would be white. So there's so many different combinations of ways to be purple or white, right? So little p, big C is white because it doesn't know how to be purple. Big P, little c is white because even though it knows how to make purple, it doesn't have permission to make purple. Okay, so what does a question look like with epistasis? Okay, so what does this look like in a question? Well, if I have a homozygous purple plant that has homozygous permission, so it is going to look like big P, okay, homozygous permission, so big C. All right, so this plant is going to be purple because it knows how to make purple and it has permission to make purple, is crossed with a plant that is white and does not have permission. So it is little p, that's white, okay, but it would also be extra, well, not extra white, but it doesn't have permission even if it did know how to make purple, okay? So diagram the Punnett square for the F1 and F2 generations and calculate the phenotypic ratios for each generation. All right, so again, these are two different genes, even though they deal with the same trait. That's why it's called polygenetic inheritance. Multiple genes, same trait, but they're different genes, so we would have to FOIL it all out, okay? Now, hopefully you guys have had a little bit of practice and you know that 100% of my offspring are going to come from these crosses. So my homozygous dominant plant can make only big P, big C. My homozygous recessive plant can only make little p and little c. So 100% of my F1 generation is going to be heterozygous for both traits. Okay? Now, what is the phenotype of this? Well, it knows how to make purple because it's got a big P, and it's allowed to make purple because it has a big C. Okay, so it is purple. That's it, it's purple. 100% of my F1, this is my F1, okay, This is my parental generation. This is my F1 generation. 100% of them are going to be purple. Okay. Now it says, what is my F2 generation going to look like? Well, so my F2 generation now is going to be 2 plants from my F1. So big P, big C, little c times big P, big C, little c. Okay, so unfortunately, we're in a state here where we have 4 different gametes that can be made here. Okay, I would FOIL out 4 different gametes that could be made here. Okay, and same with that parent. So it is going to be the dreaded 4 by 4 Punnett square, and I don't have any repeated alleles, so I have to do everything. Okay, it's one parent. Now, some of you might be thinking, well, Mr. Force J, hey man, work smart, not hard. What about the 9 to 3 to 3 to 1 ratio? Well, the problem is this is non-Mendelian. That is a Mendelian ratio. So unfortunately, I cannot use it because there's going to be only 2 phenotypes. There's only going to be purple flowers and there's only going to be white flowers. Okay, so there's no 9 to 3 to 3 to 1 because it's non-Mendelian. This is some weird genetics here that Mendel had no idea about. Okay, so here we have big P, and it's allowed to be purple. Okay, big P, and it's also allowed to be purple. Big P, little p, and it's allowed to be purple. And it's allowed to be purple. So we have 4 purple plants. P, little p. You guys are probably working faster than I am right now. Oh, these P, little p. No, sorry, this is big P. This is a big P. I can't read my own writing. I apologize. It's going to be a little c and a little c. Okay, so that does not have permission. So here we have a white one. Okay, so big P, big C, little c. And we have big P, little p, and we have little c, little c. So again, this guy, even though it knows how to make purple, it does not have permission to make purple. So that's white. Okay, so big P, little p, big C, big C, big P, big C, little p, big C, big C. So this one has permission, but it doesn't know how to make purple. So it would be white. Okay, little p, and we have a big C and a little c. So again, it doesn't know how to make purple, even though it has permission. So it would be white. Okay, big P, big C, little c. Big P, little c. So we have another white here. Okay, little p, big C, little c. So another white. And little p, little c, another white. So how many whites do we have? 1, 2, 3, 4, 5, 6, 7. Okay, so this would be a 9 to 7 ratio of purple to white. Okay, so not a 9 to 3 to 3 to 1 at all, 9 to 7. Okay, guys, this one, I want you guys to give it a shot, and then I will do it on the other slide. Okay, and but I want you guys to give this one a shot. It's the same kind of deal. Two genes that deal with coat color. That's it, just coat color. But here we have two different. Well, we have two different genes that deal with coat color, but the result is actually three different coat colors. Okay, but it's still gonna foil gametes, all that kind of stuff. All right, but we have instead of just two phenotypes, we actually have three phenotypes. Okay, so you guys give this one a shot on your own. Pause the video right now, and then when you're done, advance it, and I'll work through it on the other slide, and we'll do it together. Okay, so here we have a case where, again, we're going to have a big Punnett square to deal with. Okay, we're going to have a 4x4 Punnett square because both parents are going to be heterozygous. But we have to be careful because there's more than just purple and white like they were in the flowers. Here we have black, brown, and yellow. Okay, so we want to FOIL this out. And if we FOIL this out for both parents, right, so we're going to have a big B and a big E. We are going to have a big B and a little e. We're going to have a little b and a big E. And we're going to have a little b and a little e. Okay, and that's the same thing on this side because both parents are heterozygous. Okay, so big B, big B, big E, big E. E. Sorry guys, my pen is, or my mouse that I'm using as a pen here is not writing. I'm going to try to see if I can get a stylus or something to make this a little bit easier on me and you guys having to sit through this. But right now, it's crazy time, right? I'm just sitting here, and like I said, it's about 2:30 on a Tuesday afternoon, and they just declared a state of emergency. So my watch is going off like crazy. I'm getting all these messages. I don't know what that means. But can't be good. All right. Anyway, we're done here. So what we need to do is we need to look up here. Anytime we have a big B and a big E, that's going to be black, okay? So big B, big E, black, 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 black. So 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, brown is going to be two little B's and a big E. So 2 little b's and at least 1 big E. So brown, 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 3, okay? And then yellow is 2 little e's and it doesn't matter what else. So 2 little e's and it doesn't matter what else. So we have 1, 2, 3, 4. So a 9:3:4 ratio. Okay, so hopefully you guys kind of got something out of that. What I would like you guys to do now is to complete the questions on page 18 and 19 of your note package. Okay, if your stuff is at school, you can print the note packages off on the Google site, not the Google Classroom. This will be posted on the Google Classroom. Go on the Google site, and I will have the note packages for all the units available there. Remember, the textbook is also online, so read the textbook. As far as a quiz goes, I don't know how we're going to do that yet. I hope to have some sort of test or quiz. Most likely, it'll be a test on all of genetics. Okay, so probably after spring break sometime, we'll do a test. I don't know. You're gonna have to hand it in online, but we're still figuring all that out. I'm still kind of learning how to use the Google tools and stuff. So I'm sure I'll be able to make some sort of online assessment. Okay, but right now just practice this. Also, there's Non Mendelian extra practice worksheet on the Google site. So go on there, look for Non Mendelian extra practice. Okay, and that's what I would like you to do for the next couple of days until I post the next lesson. Okay? Stay safe, stay well, and hopefully we'll see you guys soon. Maybe some of you aren't hoping that, but it's lonely here without you guys. So take care and email me if you have any questions. Thank you.